Friends, myself Jagadish, lecturer in Zoology, KB DAV College, Nirakarpur. This time I am going to discuss about some of the PG Zoology entrance questions of the year 2021. Here the first question is, regarding the lac operon, if lactose is present, which of the following occurs? Out of these four alternatives, alternative C is the correct answer. It means lactose binds to the repressor, which does not bind to the operator, and RNA polymerase transcribes the needed genes. Let us discuss the details. When lactose is not available, lac repressor binds tightly to the operator, preventing transcription by RNA polymerase. But when lactose is available, the lac repressor does not bind to the operator. So, the way for RNA polymerase becomes clear to transcribe the operon. Next, we proceed to the next question. Pribnow box is centered at, options are A plus 10 position, B minus 10 position, C minus 35 position, D plus 35 position. Here the correct answer is B, minus 10 position. Friends, Pribnow box is a sequence of thymine and adenine nucleotides in 6 nucleotide sequence, which is the essential part of a promoter site on DNA for transcription to occur in bacteria. In bacteria, the Pribnow box is located at minus 10 position. In bacteria, the promoter consists of two short sequences at minus 10 and minus 35 position upstream from the transcription start site. The sequence at minus 10 is called the Pribnow box or minus 10 element, and it is usually consisting of 6 nucleotides, that are T, A, T, A, T. Next, we proceed to the other question. If the ratio of A plus G to T plus C in one strand of DNA is 0.70, what is the same ratio in the complementary strand? In DNA, the complementary strand contains the bases in reverse to the other strand. Here, the complementary strand should contain bases which are reverse, that is G plus T to that of A plus G, and it is reverse of the ratio of the first strand, that is 1 by 0.7 when we made solution, we will find the ratio is 1.42 point something. Hence, the correct answer is 1.43. Let us just proceed to the next question. A non-supercoiled B DNA molecule is composed of 4800 base pairs. How many helical turns are present? Here the alternatives are A 10, B 380, C 480, D impossible to determine. The non-supercoiled B DNA contains 10 base pairs per turn of DNA helix. Hence, 4800 divided by 10 equal to 480, that is 480 helical turns are present when there is 4800 base pairs are present in a B DNA molecule. Hence, the option C is the correct answer. Friends, different forms of DNA contain different numbers of base pairs per turn. The A form contain 11 base pairs, B form contain 10 pairs, C form contains 9 pairs, D form contain 8 pairs, and Z form of DNA contain 12 pairs of base pairs. The next question is, at what substrate concentration is the velocity V0 of an enzyme catalyzed reaction is 25% of Vmax. Here the options are A 3 by 4 km, B 4 km, C 1/3 of km, D 1/4 of km. Here the correct answer is C, that is 1/3 of km. Friends, according to Michael-Menten equation, V0 equal to Vmax into substrate concentration divided by Km plus substrate concentration, where Km is the Michael-Menten constant. According to the question, V0 tends to 25% of Vmax. That means V0 become 25% of Vmax, that is 1/4 of the Vmax. If we put Vmax 1/4 of the Vmax at V0, then we will find the value, that is 1/3 of km after making the solution of the Michael-Menten equation. Hence, here the answer C is the right answer. The question is, choose the mismatch. A. D-glucose and D-fructose anomer, D glucose and D mannose epimer. C alpha D glucose and beta D fructose anomer. D D glucose and al glucose are enantiomer. Here the correct answer is A, that is D glucose and D fructose anomer, because D glucose and D fructose are constitutional isomers. They have same molecular formula, that is C6H12O6, but D fructose has primary alcohol at C1 and a ketone at C2, that is carbon 2 position, but D glucose has an aldehyde at C1, that is carbon 1, and a secondary alcohol at C2. Hence, this is the mismatch found in case of A. Hence, A is the right answer. This much for today.